Welcome to another video tutorial. In this video, I will show you how we can use different online tools to predict where our desired protein is located. We know that if we know the subcellular localization of our desired protein, so it will help us to determine the function of our protein. So let's suppose if our protein is located in mitochondria, we will know that its function will be related to energy hub. And if our protein is located in chloroplast, we will know that definitely it leads to the function of photosynthesis. And some of them are located in nucleus. So how we can use online database? Today I will introduce one online database that is called TargetP2. You just need to open Google and you can write TargetP2. The first link will be of TargetP2. We know that TargetP2 serve prediction of N-terminal pre-sequences. You can just open this link. After opening this link, it will take you to this page. So here it is written that protein sequence should not be less than 10 amino acids and the maximum number of protein is 5,000. So you can just paste your protein sequence here. So let's take, for example, we can take this protein like the DPS. Let me retrieve its protein sequence. Yeah, we can download its sequence. You just need to copy and paste your desired sequence for which you want to predict the subcellular localization. You can just paste. And this is the last one is the stop codon. Maybe you can remove this. And from the organism group, we know that because this protein belongs to rice, so we can choose plant type, and you can leave this option as such, and you can just simply click on the submit. It will just take few seconds to process this job. We will know that where this protein is predicted. Yeah, here is the summary of our predicted sequence from the plant one. So we can know that the most of the protein predictions, all options are showing, but if we try to analyze this output, how we can analyze? So the maximum likelihood is other than this all. Other mean it also includes nucleus. But if we talk about like the signal peptide, that is also in 0.002, that is quite less. And if we talk about thalakite membrane, that is also in points. So these three, like the chloroplast, mitochondria, and others, like this is 0.4, this is 0.15, and this is 0.36. So there are three possibilities that this protein can be localized in nucleus, like I mean the others. And it is also possible that this protein may be located in mitochondrial and chloroplast. So in this way we can know that, but we will see another example that like here we can use another protein like this. We will try to check where this protein is located. So let me download its sequence. Yeah, here is its protein sequence. You can simply copy here. You can go back and similarly paste your protein sequence here. You can just remove this stop codon, and because this is also plant protein, so you can simply submit this. It will take few seconds to process the data. Yeah, here is the summary of predicted sequence. We can see that here the maximum likelihood is in other, Like we can see that it is strongly showing that it is not located in chloroplast, thylakoid, or mitochondrial, or even the signal peptides are, I mean, the values are 2 less than 0. So the strong chances are there that this protein is located in the nucleus. So in this way, you can use this target P2 server to predict that where your protein is subcellularly localized, either in mitochondria, chloroplast, or secretory pathway. So hope so this video will help you to determine the function of your desired protein. So if you want to know more about molecular biology and proteins, please subscribe to my channel. And if you have any other question, please let me know in the comments. Thank you for watching. Bye bye.